sexually transmitted in some cases, some types of that. All right, what's an operon? Gene plus its promoter and its terminator. In your book, they get into a whole thing. It's got an operator, then a promoter, then it's got 3 structural genes, then it's got the terminator. All you need to know for me is an operon is a gene plus its promoter and its terminator. What's the promoter for? Starts the gene and regulates expression of the gene. What's the terminator do? Ends the gene. Terminator ends the gene. Promoter starts and regulates. Terminator just ends. What do you call it if a gene that's normally expressed is shut off? Repression. I'm repressed, I'm shut off. What do you call it? A gene that's normally not expressed is in a new environment, the organism is in a new environment, new nutrient called an inducer. What does the inducer do? It turns on the gene, right? And then the gene codes for an enzyme that enables it to use the nutrient. Dokey. What's provirus? Late viral infection. Good. We'll also talk about lysogen. The word lysogen, remember I mentioned there's a lytic cycle and a lysogenic cycle. In the lysogenic cycle, bacterium is infected with a non-infectious, with a temperate, mild phage. Then it's called a lysogen. So a lysogen is a bacterium that has a prophage in it. In human cells, we call it a provirus. In bacterial cells, we call it a prophage. A lysogen is a bacterium that has a prophage. It has a temperate phage inside. We'll cover that too. But just in case you take it early, you should know that what provirus is. And prophage is the same thing as a provirus, only for bacteria. Don't forget your metric conversions. What's a milli? What's milli mean? 1,000. What does micro mean? Million. What does nano mean? Billion. Good. Okay. Difference between disinfectants and antiseptics. So easy, so easy. Disinfect the table, antiseptic on my skin. Okie dokie. You want to sanitize something? How are you going to do that? Let's say you want to sanitize the public bathroom. Wipe it down with a disinfectant. Squirt some of that blue stuff in the bowl. Okay, we already talked about this one. If you use an autoclave, it's because you want to sterilize. In order to sterilize, you have to kill everything in it, including endospores and prions, and viruses, and molds, and mold spores, and protozoa, and cysts of protozoa, and all that stuff. Alrighty, what if I treat your burn with silver? What kind of an agent is that? It's a metal, it's heavy metal. Remember your chemistry? Silver is a heavy metal, so that would be a non-competitive enzyme inhibitor. Would be toxic to those, but they do that. They put silver on burn wounds because the silver will also kill bacteria. They don't want bacteria to colonize the burn wound, so they put silver-laced bandages on it. Sometimes at work you'll see that there's ointments with silver in it, and there's also bandages with silver in the bandage.
But it would hurt.
Because the silver will prevent the microbes from growing. What?
But it would hurt, right?
I don't think it hurts the patient too much. The burn wound itself is probably what the patient's feeling. Yeah. Of course, it can if they, yeah, it can have a tendency to burn probably, but the way they make it in the bandages probably doesn't. Biosafety levels Don't forget them. Okay, biosafety level 1 is the least careful. What's your most common bacteria that we work with in the lab?
E.
E. coli. Biosafety level 1. Biosafety level 1. We're not fools. We're not going to give you anything worse than that. Biosafety level 2. Now what are you talking about? Stuff like MRSA. Stuff that's going to be more harmful. Biosafety level 3. Worried about tuberculosis. Ebola.
Ebola is 4.
Oh, Ebola's 4. Is it 4?
Mm-hmm.
Yep. So check your biosafety levels, have that handy, because we'll have a question about that. Alrighty. When I use the word antimicrobial is a general word. Antibiotics are antimicrobial, disinfectants are antimicrobial, antiseptics are antimicrobial. Antimicrobial is a general word. Disinfectants can be antibiotics or antimicrobials. A lot of overlap in these terms. Alrighty, we already talked about if you're going to test if your autoclave is working properly, what are you going to test it with? What are you going to make sure it kills?
Endospores.
Endospores, some bacillus endospores, right. And hand washing is what? Is it
De-germing.
sanitization? It's de-germing, right. And we already talked about this. Chemotherapy. Who gets credit for inventing chemotherapy for syphilis?
Ehrlich.
Right. What kind of antibiotics works well on bacterial cells, particularly because of the peptidoglycan wall? What?
Penicillin.
Beta-lactams, right. Beta-lactams work well on bacteria. And azoles, A-Z-O-L-E-S, work well on fungi and target the fungal plasma membrane. One of your patient's chart says he has beta-lactamase positive gonococcus. What's he got? He's got penicillin resistant gonorrhea. Beta-lactamase positive, Colton says, means he's got an enzyme, beta-lactamase, that breaks down beta-lactam antibiotics. Penicillin's a beta-lactam antibiotic. The bacteria has an enzyme to break down penicillin. So it's penicillin resistant. Gonococcus is gonorrhea. Alrighty. Okay, I actually did look this up and it's a test question. Penicillin, bacitracin, methicillin, vancomycin, we use those on bacterial cells because they target peptidoglycan. Tuberculosis has a lot of peptidoglycan, but the real problem is those mycolic acid waxes. The drugs can't get past those mycolic acid waxes. So what's the drug that we give for tuberculosis specifically to target those mycolic acid waxes? Remember isoniazid? I call it isoniazid, but it's actually isoniazid. It's all over your book, it's all over your PowerPoints in lab. Isoniazid, that's what they use for tuberculosis. They couple it with another drug, rifampin, which interferes with nucleic acid synthesis. If you have that chart in your textbook, chapter 10, the antibiotic chapter, it's got a bacterial cell picture, it's also in the PowerPoints. It's got a whole list of antibiotics that target the cell wall. Isoniazid is on there, but they don't mention that it's targeting mycolic acids. But that's why we use it for tuberculosis. It targets those mycolic acids, which are very waxy. TB's hard to get. All right. Targets of different antibiotics. What targets the plasma membrane?
Penicillin.
Polymyxins. What targets the ribosomes? Tetracycline and doxycycline. Why don't we want to give tetracycline to growing children and pregnant women? Great teeth, right? Okay, let's say, you took an antibiotic, and the antibiotic did its job, got rid of your pathogenic bacteria, but at the same time, it disturbed your normal flora. Give me 2 examples.
Coli, C. diff, Candida albicans.
C. diff and Candida albicans. Good. Alrighty. How does tetracycline work? You can just go through it again. Inhibits protein synthesis. Protein synthesis, all bacterial cells have to do. They all have ribosomes. They all have to do protein synthesis, so that makes tetracycline your classic broad spectrum antibiotic. And it can get a variety of bacteria, gram-negative and gram-positive. It's because it's targeting something that they both have. And they both have ribosomes. They both have the synthesized protein. Also, we talked about synergistic effects. You might get a same question on your lab quiz 5 as on exam 3. I've already noticed like 3 questions that are exactly the same on both exams. So you might get it on both exams. Because remember, Canvas kind of randomly picks what questions you get.
I think
Alright.
sometimes you get the same question in the same exam.
Yeah, I got
Yeah.
the same question in a row.
I know. You know what? I noticed that, and I didn't know how to fix it. And thanks. I'll have to ask Rich about that. I have duplicate questions
I
showed up.
find as many as 3 of the same question on the same exam.
Yeah, it shouldn't do that. But see, what happened was, well, not everybody, but a lot of the faculty at the school, we were in Angel. And then we switched to Canvas. So all our Angel materials got moved into Canvas. And when that happened, some little glitches happened. So this is my first semester in Canvas, so I'm just starting to discover them and clean them up. So I apologize. If it comes down